Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Roslin, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Leisha Kopp from Mirus Bio.
Thanks for the introduction, and thank you to everyone who's tuned into our webinar on increasing cell and gene therapy manufacturing capacity with higher titer LV and AAV transfection platforms. My name is Leisha Kopp, and I'm an application scientist at Mirus Bio. I work with researchers around the world as they evaluate and adopt Mirus technologies for use in their own experiments and processes. And I look forward to sharing more about our products and strategies for developing effective systems with you today. We'll start today's talk by answering the question, what makes Mirus biotransfection solutions unique? We will then cover critical parameters Mirus used to develop our LV and AAV kits, as well as some key considerations for choosing transit Virusgen for your viral vector platforms, whether you're producing lentivirus or adeno-associated virus or working with adherent or suspension 293 cells. Following this presentation, we will have 3 quick polling questions for you, and then we'll open the floor for Q&A. In conversations with researchers, we often get the question of what makes Mirus reagents different, as well as why can I expect better results if I switch to your products? Both are fair questions that I'm happy to address. Because while many transfection formulations utilize a cationic polymer like PEI or liposomes like lipofectamine, Mirus formulations are non-liposomal mixtures of both cationic lipids and polymers, which promote effective complex formation to a broad spectrum of cells. Our formulations tend to be less toxic than liposomal formulations because they're less disruptive to the cell membrane. And unlike PEI-based formulations, Mirus reagents are effective for delivery of multiple nucleic acid types to a wide range of cells from many different applications. I'd also like to add that Mirus strives to provide the best support possible for our customers. Our scientific team is comprised of world-class chemists and biologists committed to finding those perfect formulations and protocols to streamline and improve transfection-mediated applications. We also offer a technical support team of R&D and application scientists that work closely with the reagents you're using or are interested in using. This means that when you reach out to us via phone, email, or web chat, you immediately reach an expert that can help address your questions on the spot. In addition to effective products and dedicated support, Mirus also offers 25 years of nucleic acid delivery expertise. We're here to talk about the transit Virusgen kits and reagent today, but we also offer products for general DNA delivery and DNA RNA co-delivery, products for specific applications such as viral vector production and protein production, as well as products that utilize alternative strategies for transfection such as electroporation, all with the goal of supporting delivery of any nucleic acid to any cell type to support all cell biology applications. And in our 25-year journey, we've made some friends around the world. Mirus has over 30 trusted distribution partners supporting over 40 countries in all major markets. If you're outside the US and are interested in which companies distribute in your area, you can find our list of international distributors at mirusbio.com/contact/distributors. We also offer international direct ordering to countries for which we do not currently have a distributor. Now that you know a bit more about Mirus Bio, let's take a closer look at products and protocols optimized for higher titer lentivirus and adeno-associated virus, or AAV, production. I'm sure it's no surprise that supporting cell and gene therapies was top of mind when developing a new transfection formulation for LV and AAV production. As we all know, lentivirus is often a critical component of cell therapy, as shown in this example, where patient cells such as T lymphocytes or natural killer cells are harvested and then genetically engineered to express a protein such as a chimeric antigen receptor or a CAR that better enables the cell to detect and eliminate cancer cells once expanded and re-administered into the patient's body. Adeno-associated virus is commonly used in gene therapy, where the virus itself is the therapeutic as it contains a corrected copy of a gene which can be directly administered to the patient. Currently, there are 3 cell therapies and 2 gene therapies approved by the FDA. In 2017, Novartis's Kymriah, which is designed to treat acute lymphoblastic leukemia, and Kite's Yescarta, which is designed to treat non-Hodgkin lymphoma, were introduced. Very recently, in July of 2020, the FDA approved Kite's Tecartus for treating relapsed or refractory mantle cell lymphoma. On the gene therapy side, in 2017, Spark Therapeutics launched Luxturna, which is a gene therapy directed at correcting inherited retinal disease due to mutations in RPE65. In 2019, Avexis introduced Zolgensma, an AAV-based therapy designed to treat spinal muscular atrophy. Each of these therapeutics is groundbreaking and life-changing, and according to recent reports from sources such as BioProcess International, there are more than 1,000 cell and gene therapies in development in 2020, with the potential for greater than 10,000 cell and gene therapy clinical trials by 2026. And of the therapies being explored, over 60% utilize either LV or AAV for gene delivery. So clearly there's a need for cell and gene therapy workflow support, and that's what leads us to develop the Transit Virusgen reagent. When designing a reagent to support cell and gene therapy development, we had several goals in mind. First, we knew that we needed a reagent formulation that supported delivery of multiple plasmid DNAs to HEK cells, which is the basis for both lentivirus and AAV generation via transient transfection. Beyond that, we wanted to develop a reagent that was best in class, so to speak, for not just LV or AAV, but for both. Secondly, we screened libraries of our lipids and polymers for a combination that promoted effective complex formation for both adherent and suspension cell types, as this formulation would then be able to support large-scale LV and AAV production in formats such as the iCELLis and roller bottles, as well as stir tank bioreactors. And last but not least, we knew we needed a formulation that supported reproducible and scalable virus production. Ultimately, our goal was to simplify the viral vector development process by delivering a formulation that yielded more functional virus per transfection to streamline processes and reduce production costs, and Transit Virusgen is able to do those things. The features of our Transit Virusgen formulation include superior performance, cell line and platform flexibility, batch-to-batch reliability, and in addition to a functional QC, Virusgen Select, which is shown here, includes release testing and quality documentation to streamline the process of raw material qualification to ensure a seamless transition from discovery through large-scale manufacturing. Transit Virusgen is chemically defined and animal origin-free, and there is no commercial license required for R&D or manufacturing use. PEI-based reagents are the most commonly used formulations in viral vector manufacturing, which is really what set the bar for reagent development at Mirus. This data, generated in suspension 293F cells and freestyle F17 media, demonstrates that Transit Virusgen outperforms both 25 kilodalton PEI and PEI Pro from Polyplus by generating up to 10-fold higher functional titers for lentivirus and up to 4-fold higher titers for AAV production. Virusgen, as demonstrated in this figure, also strongly outperforms PEI formulations in adherent 293 T cells grown and transfected in DMEM plus 10% FBS. For more details about these experiments, please visit www.mirusbio.com/select. In these next few sections, we'll look more closely at critical workflow parameters that impact lentivirus and AAV expression levels, as well as how these factors guided development of our LV and AAV kits and recommended protocols. This figure depicts the process we followed when developing the Virusgen LV and AAV kits. The first step was scouring the literature as a starting point for developing candidate compounds. We then tested each of these potential enhancers for performance in our established lentivirus and AAV workflows with Transit Virusgen. With the most promising candidates, we investigated other critical parameters, including DNA amounts and cell density at transfection for both adherent and suspension cells. And with our final LV and AAV systems, we also compared expression levels with various sets of plasmids, various serum-free cell culture medias, as well as different suspended 293 cell types. The first critical component that we explore are the impacts of total DNA and cell density at the time of transfection. These figures show a DNA titration from 1 to 2 micrograms of total DNA per mil of culture for both LV on the left and AAV on the right. We found that lentivirus production without the LV enhancer, the black bars, is higher when using 1.5 to 2 micrograms per mil of total DNA. However, only 1 microgram of DNA is needed when using the LV enhancer. This data also shows that the LV enhancer boosts titers 3 to 5-fold over reagent alone conditions. For AAV, we found that 1.5 to 2 micrograms of total DNA per mil of culture is optimal with and without the enhancer, and that the optimal DNA amount will vary depending on the serotype and vector sets used. As shown by the red bars at right, the enhancer doubles AAV production in all conditions. This does bring up an important point for platform development, as the total DNA required for optimal expression may depend on the interactions of several components, and for this reason, we highly recommend running a DNA titration with your final vectors, cells, and media to determine the lowest DNA amount you can use for robust expression. Cell density at transfection is another commonly explored parameter. For lentivirus and AAV production with transit Virusgen alone, meaning without the enhancers, 2 million cells per mil at transfection was best. In contrast, significantly higher titers are obtained when using the LV enhancer, the red bars, at 4 million cells per mil. For AAV, we see equivalently higher titers at 2 and 3 million cells per mil, both with and without the AAV enhancer. Again, it's important to do a cell density titration for your final cells, gene of interest containing vectors, and media to determine what is optimal. Based on the data we just covered for DNA and cell density at transfection, this slide depicts the final LV and AAV workflows. For the Virusgen LV kit, the day before transfection, cells should be split to ensure that they are rapidly dividing at the time of transfection. On the day of transfection, dilute cells to 4 million cells per mil, form transfection complexes per the protocol by combining 1 microgram of total DNA and 3 microliters of transit Virusgen per 1 mil of culture. Incubate for 15 minutes, add complexes to the cells, and place cultures at 37 degrees, shaking overnight. 18 to 24 hours post-transfection, add the LV enhancers to the culture at 10% of the total culture volume. At 48 hours post-transfection, harvest the lentivirus from the supernatant and assay as desired. For AAV, the upfront work is similar, however, cells should be diluted to 2 million cells per mil for transfection rather than 4 E6 per mil. Also, the AAV enhancer is included in the complex formation solution, so there's no need to reopen the culture post-transfection. At 72 hours post-transfection, or whatever time is best per your AAV serotype, harvest and purify or assay is needed. I do want to pause a moment on the complex formation part of this process. This can be a pain point for large-scale LV and AAV production workflows when longer periods of time beyond that 15 minutes specified by our standard protocol for complex formation are needed for preparing and adding complexes to cells in bioreactors. Here we compare two commonly used complex formation methods and their stability over time. For complex 1, which is the red bar, transit Virusgen reagent and the AAV plasmids were diluted separately in PBS, each at 5% of the total culture volume. Following a 5-minute incubation, the two tubes were combined and mixed thoroughly, which starts the complex formation process. Aliquots of the complex were added to cells at either 15, 30, or 60 minutes post-initiation of complex formation. In contrast, complex 2, depicted by the orange bar, was formed by diluting transit Virusgen in PBS, waiting 5 minutes, and then adding concentrated DNA to the Virusgen PBS tube, followed by a quick trip to the vortex for mixing. These complexes were also added to cells at 15, 30, and 60 minutes. What this data shows is that both methods work equally well and that you can expect similar titers of adding complexes anywhere from 15 to 60 minutes. Using the protocols outlined a couple slides ago, we again compared the performance of Virusgen as well as the Virusgen LV and AAV kits to both 25 kilodalton PEI and PEI Pro. in suspension 293F cells in freestyle F17 media. With the revised protocols and enhancers, our improvement over PEI-based methods is another 2 to 3-fold greater than what was achieved with the Virusgen reagent alone. The next critical component we would like to take a closer look at is vector design. Until recently, most of our Virusgen reagent and LV kit development work was done with 3 different packaging plasmid mixes, the first being the 3rd generation virus-safe lentiviral packaging plasmids from Cell Biolabs. The second was the 3rd gen hybrid mission lentiviral packaging mix from Sigma, and the third was the 3rd gen LV mix packaging plasmids from Thermo. Traditionally, we saw highest titers using the mission packaging mix, which isn't unexpected, as there are fewer total plasmids to transfect with that 3rd gen hybrid system. However, more recently, we tested the P-ALD packaging, LV packaging, and transfer plasmids from Aldevron, and honestly, we were very impressed by their performance. Here we see functional LV titers nearing 1 billion TU per ml, which really raises the bar for commercial plasmids and also begs the question, what else does this improve? Do these types of vectors enable the use of less DNA in reagents? In this next experiment, we transfected 293F cells at 4 million cells per ml with total P-ALD vector DNA ranging from 0.5 micrograms per ml to 2 micrograms per ml with transit Virusgen and the Virusgen LV kit. The black bars show titers without the LV enhancer, and we see that while titers are still very high with 1 microgram per ml, they do increase when using more DNA. However, when adding the LV enhancer post-transfection, the highest titers are achieved when using 0.75 to 1 microgram total DNA per ml. This is another nod toward doing a DNA titration with your final system, as less DNA may be required when all components are optimized. In this lentivirus expression experiment, we used the P-ALD packaging vectors from Aldevron and a transfer plasmid expressing GFP to compare performance of the Virusgen LV kit to PI Pro and PI Max. XB293 cells were adapted for several weeks prior to transfection into the XB293 expression media, freestyle F17 expression media, LV Max production medium, and the balanced CD HEC293 medium from Irvine Scientific. Cells were then transfected with 1 microgram total DNA at 4 million cells per ml. We found that the Virusgen LV kit yielded 3 to 7-fold higher functional titers as compared to PI Pro and PA Max. I'd also like to note that we've repeated this experiment several times, and that trend is very consistent. Another trend we see is overall highest LV expression with the balanced CD media and XB293 medias, with slightly lower titers consistently observed in freestyle F17 and LV Max. We performed a similar experiment using XB293 cells adapted to the same 4 medias. However, this time for AAV GFP expression using the AAV helper-free system from Agilent Technologies. Transfection complexes were formed with 1.5 micrograms total DNA and 3 microliters Virusgen reagent per ml in the AAV complex formation solution and enhancer. Results show up to 17-fold increase in AAV titers over the PEI-based transfection formulations when using the Virusgen AAV kit. And again, we see highest overall titers with the balanced CD293 and XB293 medias. Now that we've gone through our established LV and AAV kit workflows, and more than a slide or two of data, let's talk about what higher titers really mean for your viral vector platform. If we take a step back and look at the requirements for effective cell and gene therapy doses, the need for better expression platforms becomes very clear. The average cell therapy requires between 10 to the 8th and 10 to the 10th viral particles per patient. For cell therapies, virus is used to transduce cells ex vivo and therefore will never need to contend with the patient's immune response. For gene therapy, however, the average patient dose requires 10 to the 11th to 10 to the 16th viral particles to be effective. This is because the virus is directly administered to the patient and must affect the entire target organ or system. In addition, the virus will contend with the patient's immune response. If we do the math and translate viral particle requirements to the amount of cell culture required to produce that much virus, it becomes very evident that the higher titers are important. So let's do that math. Let's say that A locally administered gene therapy dose requires 3 times 10 to the 11th viral genomes, and a systemic dose requires 5 times 10 to the 15th viral genomes. We get roughly 3 times 10 to the 9th VG per ml from PEI-based production, and assuming even just 4 times higher titers, we would get 1.2 times 10 to the 10th VG per ml with transit Virusgen. If we multiply that out, we find that greater than 1,600 liters of AAV culture is needed to generate 1 systemic patient dose using PEI. When using Virusgen, that required culture volume per systemic dose drops to just over 400 liters. If we think about what higher titers mean on the production side, it's a significant reduction in material requirements when fewer bioreactors and far less media and costly GMP-grade DNA is needed to generate a sufficient amount of virus. It's less hands-on time, less paperwork, less time in expensive manufacturing suites. And in summary, higher titers mean more efficient use of time and materials and an overall reduction in workflow costs. To show this in a different way, higher functional AAV and LV titers lead to lower production costs, more efficient use of time, which ultimately leads to more patients treated. As I mentioned earlier in the talk, Virusgen is designed to support you from R&D through commercialization. Our research-grade product is configured in vials of up to 1.5 mils and is sterile filtered and functionally QC'd. Transit Virusgen Select is designed to support preclinical and early phase clinical trials and is dispensed at 30 mL per bottle. Assays for sterility, bacterial endotoxin, mycoplasma, and formulation identity are performed per USP guidelines on Virusgen Select, and they are reflected on the C of A, which you can download a copy of from our website. The Virusgen GMP reagent will be available in early 2021, along with the LV and AAV enhancers. Virusgen GMP will be a 150 ml dispense, which is sufficient for about 50 liters of transfection, and will also undergo the same quality release testing as is performed on the Transit Virusgen Select product. All Virusgen reagents are identical in formulation, ready to use, chemically defined, and animal origin free. If you are interested in testing either the Virusgen reagents or Virusgen LV or AAV kits for evaluation in your viral vector production platform, we definitely want to support that. Please visit our sample request portal at mirusbio.com/sample and choose the Virusgen Select from the large-scale protein or virus production section. Once we receive and qualify your request, we will contact you to verify which sample you'd like. If you are beta testing the LV or AAV kits, we will arrange a prep talk with you prior to your evaluation of the kit to walk through the protocol and address any questions that you may have. You'll then receive and evaluate the sample. Please note that we are here to assist with any questions you have during that time. So if something isn't going well, if the protocol isn't making sense, you can always reach out to us. We are definitely here to make sure that things are clear. Following your evaluation, we ask that you send us feedback. Now, you do not need to share any proprietary information, and if you're more comfortable discussing feedback with the CDA, that is also fine. We can get that in place. However, we do want to know your thoughts on the product, on the protocol, and overall performance of Virusgen compared to your current system. And hopefully that leads to adaptation or adoption of our system in your workflow. If you'd like to learn more about Transit Virusgen, there are a number of resources at your disposal. The Transit Virusgen Select page is shown here, as is our sample request portal. As you can hopefully see by the tiny writing here, Virusgen Select is circled in red. There are also webinars and video tutorials available for you along with frequently asked questions. You can also download the Virusgen brochure and documentation from our website if interested. To summarize, the Virusgen reagent and Virusgen LV and AAV kits were designed to offer superior performance, flexibility, reliability, and quality to cell and gene therapy researchers. Virusgen is also AO-free and requires no commercial license for R&D or manufacturing use. We sincerely thank you for taking time to learn more about Mirus Bio's Transit Virusgen reagents and prototype Virusgen LV and AAV kits. You're welcome to contact me at any point with questions at the email address listed here, so leisha.kopp@mirusbio.com. You can also contact our tech support team via phone, email, or live chat on our website. We do have a couple polling questions for you now, and then we look forward to further discussion during Q&A.
Okay, great. Thanks, Leisha. So we do have those polling questions, and you should see them on your screen. If you can go ahead and use your mouse to make the selection. What do you consider the biggest hurdle for developing large-scale viral vector production platform? Scalability, expression variability, cost of materials and labor, inadequate viral vector expression levels, meeting accelerated timelines, or I'm not currently developing a therapeutic. And the next question is, what transfection complex formation time do you require in your large-scale LV or AAV manufacturing process? No time limitations, more than 15 minutes, more than 30 minutes, 30 to 60 minutes, or more than 60 minutes. And finally, at what phase in your cell or gene therapy pipeline do you require GMP-grade transfection reagent? Select one. Process preclinical studies, phase 1-2 clinical trials, late phase clinical trials, commercialization, or I'm not currently developing a therapeutic. Go ahead and make your selections. That will help our speaker. Just going to give you a couple more seconds to do that. And then we'll start with your questions. Okay, so Leisha, the very first question is, you showed a complex formation time course out to 60 minutes. Have you tested longer complex incubation times?
That's a great question. So thank you for asking. We have done minimal testing beyond these 60-minute time points. And so I don't want to draw any strong conclusions on whether or not that is going to be fine. We do know that complexes can be very stable out to 60 minutes. One caveat is they shouldn't be mixed during that 60-minute incubation time period. They should be just left, static sitting there. It can be at room temp or at 4 degrees. That's all fine. We do know that if it's a very long time point beyond the 60 minutes, so say up to 4 hours, we do start to see a decline. And so we're actually in the process of trying to determine exactly where our limits are for perfectly stable complexes, and then what marginal decrease we would expect to see at certain time points. But up to 60 minutes, we do know that things are very stable.
Okay. And Leisha, we did want to share this survey with the audience. It looks like most people have answered, so I'm going to go ahead and do that now. If you like to see.
All right, hopefully everyone can see. I can see the results on my end here too. And it looks like, okay, so the first question was, what do you consider the biggest hurdle for developing a large-scale viral vector production platform to be? And it looks like 50% of the audience cited that scalability or expression variability was the biggest hurdle. And that's something that we hear very commonly on our end, as well as troubleshooting with folks and helping them figure out how they can scale from the smaller formats to larger. The second most cited answer was cost of materials and labor, which is another huge concern and payment point that we hear. So that all makes perfect sense. For the next question, it looks like, what transfection complex formation time do you require in a large-scale process? And the majority of people cited 30 to 60 minutes. So I think that's a great thing to see. No one actually noted greater than 60 minutes. So based on our results, the Virusgen formulation and complex window should be sufficient for most, which is great. The last question is, at what phase in your cell and gene therapy pipeline do you require a GMP-grade reagent? And 50% of our respondents noted phase 1-2 clinical trials. So actually pretty early in the process. I would say in conversations, we see that it's definitely preferable to have earlier in the clinical trial phases, but absolutely required in phase 3 and beyond. And so that was actually the second most common answer. So that all lines up very nicely. So thank you very much for your responses. Lee, I don't know if there are any other
There
questions?
are some other questions. Sorry. So what AAV stereotypes have been tested with your system?
So we at Mirus have done most of our testing with AAV2. So we have an AAV2 GFP, which is very nice for doing the functional readout. But we have customers and collaborators that are testing, that are testing stereotypes ranging from AAV2, 5, 6, 8, and 9 are the ones that I see most often. And we know that it works well in all of those so far.
And do transfection complexes have to be 10% of the culture volume, or is 5% volume also okay?
Great question. So we see people, the most common range for complex formation volumes would be within that 5 to 10% of the total culture volume. Our protocol is written for 10% of the culture volume, but we've also done pretty extensive testing using 5%. We kind of refer to those as 2X complexes because the active ingredients, the DNA and the reagents are, twice as concentrated within that matrix. And we find that that also works well out to about that half-hour point. It's something that we do want to look at a little bit more extensively for those longer complex incubation periods, because if you think about how complexes form with negative and positive charges, if they're more concentrated, they're going to form more quickly. And so we still need to establish the length of that window, but say you only need a half hour, that should be also perfectly fine to do.
Okay. Well, it looks like we're just about out of time, so we have time for just one more question. If we haven't gotten to your question, we will be passing those on to Leisha, and she can follow up with you directly, so continue to type in your questions. But the last question for the webcast is, what is the lead time for beta testing the AAV kit?
Excellent. So really there's no lead time. Sometimes it takes a bit if we do ask people to review our label license because the enhancer kits do contain a prototype. And that is just an email acknowledgement, unless, of course, your company would require a CDA. And sometimes, just the legal stuff takes a while, but in terms of just us being able to get in touch with you, set up that prep talk, and go through the protocol, and then send you material, that can all happen typically within a week.
Okay, great. Thanks, Leisha.
Thanks, Leah.
And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website. And as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.